Okay, unfortunately, before we get into cells and looking at various cells and a lot of cells in the lab, you'll be staining your own cells and looking at them in images of plant cells. We've got one problem. Cells are tiny. Most cells, almost all of them, you cannot see with the naked eye. So we will need to be using a device, a piece of equipment that you're probably already familiar with, but we just want to reintroduce, remind ourselves of. And it's perhaps in the world of biology one of the most important tools we'll use all year, and that is this guy, the compound light microscope. And we just want to use this short video to introduce some of the main ideas and concepts of this. The fact that it is, when we say it's a light microscope, well, that's pretty obvious, because what we're going to see is that you have to have a light that will shine beams of light up through an image that we'll see here. And if you remember, there's my creepy little eye. We'll be looking through this lens here, and that will allow us, through a series of lenses here, and up here, to actually magnify this image that we can see. So the light component is pretty obvious. The compound aspect of that is also rather simple, like a compound word made up of more than one part. Like, for example, firewood. It's fire is a word, wood is a word, but you put them together, it makes up a different part. So this guy here is actually made up of multiple parts and multiple lenses. It's not just one lens. Like we said, we'll have lenses here called objective lenses. We'll have lenses there called ocular lenses as well. So what we want to do is look at some basics of microscopes, and then we'll start using them in the lab. So just the name implies, micro, tiny, scope, meaning that we can look through that and see that. So obviously they're used to magnify very small objects, things we would not be able to see with the naked eye, or barely be able to see. It'll enlarge them, we can see a lot of detail. And as we said, there will be two types we're interested in here. Primarily, the one that we're really want to focus on, is the one we'll be using, is the light microscope. The other one, an electron microscope, is fantastic, but it's a little too pricey and large. It's a little out of our price range. So we want to focus on this guy right here. So let's just briefly want to show you what an electron microscope is, what it looks like, and why it's so amazing. Unfortunately, I mean, what the heck are we looking at there? Is that some Star Wars futuristic image, or what is that? That is an electron microscope. If you look closely right here, you can see binocular lenses. This is where the person would look through. And this large entire apparatus is the electron microscope, hooked up to computer monitors for imaging. It's pretty complex. Hundreds of thousands, if not millions of dollars. And it works a little bit differently in that it aims a beam of electrons on, or in some cases through, an object, a specimen we're trying to look at. So it's not going to use light, it will use beams of electrons. But what makes that so amazing and great is that we can enlarge images thousands of times, up to hundreds of thousands of times. You can take something that's amazingly tiny and make it appear 20,000 times larger. That's pretty impressive. Imagine, to put that into scale, into perspective, imagine being able to enlarge yourself 20,000 times. You know, if you're 5 feet tall times 20,000, you're going to be pretty large. A lot of detail there. The downside is that, yeah, it's so expensive, takes up a lot of space, but also you lack the ability to look at a living specimen. By bombarding it with electrons there, it actually kills the specimen, so you can't observe living organisms moving around. That's a major drawback. So we sacrifice life for detail, and so I mean, you got to kind of pick your poison. Depends on what you're looking at there. But just to show you some examples of what they look like, these are, remember in our video 1 we saw cilia. They look like little hairs on the outside of a paramecium cell. And you can look at the detail here. It's three-dimensional, it appears to be. It gives you a lot of details and shadows, and you can see that it zooms in so much. That other image we looked at with the paramecium didn't have a lot of detail. We could barely see the cilia. Here it almost looks like 70s shag carpeting. It's amazing. And we actually have two kinds, an SEM, or scanning electron microscope, or a TEM, transmission electron microscope. One scans the surface, one transmits or sends electrons through. And in essence, you get a cross section of them. This looks probably not as nice to most of you here, but here you can see the inner workings in even more detail. We can actually see parts of the cells in here. It's just pretty nice. But either way, bottom line, we get a lot of detail there. Two more quick images. These have been adjusted through computer software, but here's not magnified to a great amount, but look at the detail. This is an ant holding on to a microchip processor. You can look at the detail. It almost looks fake. It's amazing. Now again, it has been color enhanced through computer software, but you can see a massive amount of detail. And these, what do you think those are? If you look at them, they look like, what are they, aliens? Are they diseases, viruses? These are actually mixes of pollen grains. Now, when we say pollen grains, I mean, as we know, oops, going back here, when we do say pollen grains, remember pollen is simply just the sex cells, the gametes of plants, of sexually reproducing plants. That they release, like pine trees release pollen. Many plants release them. These are the sperm. They fertilize the eggs of the plants. If you look at them, that's what they look like under the microscope. It's amazing. If you think about that and looking at the fact that if you have an allergy to pollen, wow, that's what you're breathing in is kind of disgusting. But at any rate, we're able to see a lot of detail, and that's a very, very good thing. But we won't be using those. We'll be using compound light microscopes. And as we said, made up of more than one lens, and they use light. These look very much like the ones we'll be using in here, which is nice. And again, as we said, they shine light from a lamp up through a slide. We put a specimen on there and allows us to see it. We're a little bit limited in our magnification. It can only enlarge objects about 40 to a maximum. In fact, most of our microscopes won't even go this large, 1,000 times. But it's still pretty good, a lot more powerful, much, much more powerful than our eyes. And this does allow us to observe living specimens. So we might get lucky and be able to see a microscopic organism swimming around in pond scum. Disgusting, but yet oh so awesome at the same time. One small drawback, though, is many times we have to stain objects, where we have to actually apply stain to a slide, which we'll be practicing this in class, so that we can see them better. Because remember, most cells are made up of water. Water's clear and colorless, so that presents a problem. But if we apply a stain, it darkens it and we can see more detail. So what we want to do eventually in class is we will be looking at a wide variety of cells, and we can see here that with the unaided eye, this is nice because it shows you a range in scale of what you're able to see. Humans, obviously, you can actually see some large nerves and muscle cells with the naked eye. Chicken egg we'll doing a lab with. That's actually technically is a cell. Frog eggs we can see. But most plant and animal cells, and even parts of cells in this shaded range, you can see with a light microscope. Anything smaller than that down here, very small bacteria, viruses, even some parts inside of a cell. You can see proteins and lipids here. You cannot see with the eye. You cannot see with a microscope. You have to use electron microscopes. So as you get smaller and smaller, you run into some problems. You can see even electron microscopes, you can't see really atoms per se. So and see molecules, so you're even limited there. But what our ultimate goal is going to be is that we need to know and to learn through activities in labs the parts of the microscope and what they do and how they work, because we've got to be really, really specific here in doing so. Because each of them has a specific part. So really quickly, we can see here, you will see these focusing knobs. That is, a coarse will move it around a lot. Fine will move it just a little bit and help bring it into focus. But that's going to allow you turn these back and forth, and they will adjust and move either the nose piece up and down, or they'll move the slide up and down and allow you to focus the image. So that's a good thing. Our eyepiece lens right here, we do have a lens in here. It is designed to, just like looking through glasses, that's going to magnify what we're looking at. Whatever we see coming up this tube, if you will, it'll magnify it 10 times. So that'll help enlarge the image. And perhaps most important aspect here are the objective lenses. Some will have 2, some will have 3. You can see some are small, some are shorter, and usually what will happen is that the smaller lenses will magnify about 4 times, medium size will do about 10 times, and then these you'll have anywhere from 30 to some cases will even go as high as 100 times larger. So think about it. You're actually magnifying an object twice. If we have some kind of little object here we're trying to look at, we magnify it here, let's say 10 times, plus up here 10 more times. So you're magnifying a magnification. Kind of nice. The other few other things here, just really quickly with details, that we do have to have some type of a lamp or a light that we can turn on. There'll be usually a little switch here to turn that guy on. And there also is something right here that's not labeled. That will be called, the generic term for that is a diaphragm, and that will open and close and allow either more light or less light through for the object. So it has some other parts that we'll learn through activities, but the basic parts that we are responsible for knowing and properly handling and using these, because if we can use them properly, we'll be able to see a lot of different types of cells in a lot of detail. Great detail. And that'll be a great thing for us. So thanks for watching. Now let's get down to playing around with these things.